Welcome friends. Now we will talk about apoptosis. What is apoptosis? It is a program cell death which is occurred in a programmed manner. Okay. So cell death is two type. Generally, cell death is two type. One is programmed cell death, which is occurred in programmed manner, which is called apoptosis, and the second one is accidental, which is called necrosis. Now, come to the point apoptosis. What happened here? The cell which will be die is signaled. Finally, first one is signaling by killer cells protein. Then cell will be shrinkage, and the nucleus or nuclear fragmentation will occur here, and the small fragments and phagocytosis of the cell. Okay. Now come to the point. What happens in necrosis? Necrosis, in case of necrosis, cell swelling is here, and then cell burst, and cell material become causing pain for nearby. That means when in case of necrosis is occurred, the cell swelling is occurred first, then the cell will be burst because of swelling, and cell material become causing for pain of nearby. That means the cell materials such like histamine or like that, they will cause pain, which is in the cell. After burst of the cell, they cause pain. They become out from the cell. So thus they cause pain for nearby. Okay. Now come to the event point 1 of apoptosis, the cell which will be apoptosized. What will happen in this case? Phosphatidyl serine become outer surface. Okay. Phosphatidyl serine, which is generally occurred in situated in the inner surface of the cell, of the inner membrane, it will expose to the outer surface. It is negatively charged, so it will situated in the inner surface of the cell membrane. Okay. Phosphatidyl serine, which is exposed to the outer surface, is tagged with the annexin 5, which causes the eat me signal to the other phagocytes, and the cell will be phagocytosis. In this case, no pain will gain, but in case of necrosis, there will be pain, but in case of apoptosis, no pain will gain. Okay. Now come to the next point. Apoptosis is generally two types. First is generally apoptosis is two types. Intrinsic pathway of apoptosis and the extrinsic pathway of apoptosis. Now we will talk about the intrinsic pathway of apoptosis. At first, we will talk about the structure of the mitochondria. Okay, we will discuss about the mitochondria in shortly. So generally, the inner membrane of the cristae, inner membrane of the bacteria is generally this type of structure is called cristae. They are made up of two type of structure: outer membrane and inner membrane. Outer membrane comes from the eukaryotic cell, and the inner membrane is generally a bacteria. Generally, in case of evolution, bacteria at first was individual bacteria. Mitochondria is individual bacteria, but in case of endosymbiosis, laterly they have lost their some features, and the outer membrane, the covering membrane, is come from the eukaryotic cell. Okay, and they are dependent on some types of cell material product of the eukaryotes. So the inner membrane comes from the bacteria, and the outer membrane comes from the eukaryotic cell, and between them there is a intermembrane space. This is matrix, and the small points are cytochrome C, which is situated in between the intermembrane space, outer membrane and inner membrane. Okay. Come to the point, BCL2 proteins. What is BCL2 proteins? BCL2 proteins means BCL2 proteins which generally help in the intrinsic and extrinsic apoptosis. And the full form of BCL2 proteins, actually generally two types of domain here. Generally, four types of domain. Domain BH1, BH2, BH3 and BH4. BHD is generally for BCL2 homology domain, BHD. This is the anti-apoptotic apoptotic BCL2 protein and the pro-apoptotic BH1 to BCL2 protein. Okay. So come to the point 1. Generally 4 types of domain are here, but in case of pro-apoptotic BH123 domain, here we will see that 1, 2 and 3 domains are present, but 4 is absent here. Okay. It will help to form apoptosis. So it is called pro-apoptotic BH123 proteins. Now come to the second one. This is the BCL2 pro-apoptotic protein example. Example is Bach's and Bach protein is generally example of pro-apoptotic BH123 protein, BCL2 pro-apoptotic protein, that means BH123 proteins. And the anti-apoptotic BCL2 proteins which suppress the apoptosis process is generally have 4 type of domain, 1, 2, 3 and 4. They have, the examples is BCL2 and BCLXL. BCLXL and BCL2. But the BCL2 pro-apoptotic protein is Bach's and Bach. Okay. Now, here the function of Bach and Bach's protein. The Bach protein, Bach and Bach's protein generally in the outer surface, cytoplasmic space, and Bach protein situated in the membrane. So Bach and Bach generally come together to close, forming a channel. Okay. When they come together, they form a channel. Generally, Bach's protein is situated in the cytoplasmic space, and the Bach protein is generally situated in the membrane. So, cytochrome C, which is in intermembrane space, they come outside from this channel when Bach and Bach's proteins are aggregate together to form a channel. Otherwise, they can't go outside the cytochrome C. Pro-apoptotic BH3 only protein. Pro-apoptotic BH3. Pro-apoptotic, that means they help in the apoptosis. Okay. BH3 only protein, that means they have only one domain, that is BH3. But generally we know BCL2 proteins have 4 general domains. Varying these domains, there are 3 types of domain, 3 types of BCL2 proteins, that is pro-apoptotic BCL2 and anti-apoptotic BCL2. Okay, pro-apoptotic BCL2 is 2 types generally here. The Bach and Bach's protein is an example. They have generally 1, 2, 3 or BH3 only protein which helps in the pro-apoptotic here like that. And one type of anti-apoptotic BCL2 protein like BCL2 and BCLXL. Okay. Now, pro-apoptotic BH3 only protein, examples of that, these BAD, BEAM, BEAT, PUMA, NOXA, they bind with anti-apoptotic BCL2, and sequestered it. Okay. Anti-apoptotic BCL2, that means BCL2 and BCLX. They bind to the Bach's protein, generally anti-apoptotic BCL2 and BCLX. They bind to the Bach's protein and stop the forming channel form. But when there is a requirement of apoptosis, BH3 protein like beam, bead, tumor, doxa, they come to bind with anti-apoptotic BCL2 protein and sequester it. So Bach's protein is free and they form channel with Bach protein, and cytochrome C is from outer side, come to the outer side, and the apoptosis is started. Now come to the mechanism of intrinsic pathway of apoptosis. Here, DNA damage. When if there is it is a DNA actually, and when we give the X-ray, it will break. It will cause damage, DNA damage, and double strand break will occur here. After suddenly double strand break, the ATM and ATR, which means ataxia telangiectasia mutated and ataxia telangiectasia RAD3 related protein, they become activated by serine threonine kinase, and ATM ATR is phosphorylated. The phosphate group, ATM ATR phosphate group add the CHK protein, or means checkpoint kinase 1 and checkpoint kinase 2 CHK protein. The CHK1-2 protein add protein to the P53, which is the main decider of the apoptosis. They may cause DNA repair. They may control P21, which cause arrest of the cell, or the pro-apoptotic protein like Puma, Noxa. They come to the promoter region, bind 4. Then at first decide the DNA may be repaired or not. If they cause, they can be repaired, so that will happen. If not, then the cell cycle will be arrest. If much longer time they take, that means if they seated on the on promoter region so many times, much long times, so and sees that there is no type of repair or arrest, repair is doing here, so they forward to the DNA to the to be apoptosis, and the pro-apoptotic protein Puma, Noxa, they will be activated, which are BH3 only proteins. Okay. Generally, there are 53 control mechanism. When it will be add phosphate group, but means CHK1, 2, it will add in phosphate group to the P53. When it will be in single 3, that means. But generally MDM2 is added with it, P53. When ubiquitin, when apoptosis will be done, then P53 will ubiquitin will be sequestered the MDM2, okay, and P53 will be free. Now the main mechanism, anti-apoptotic BCL2 protein, okay, Puma and Noxa, BH3 proteins. This is the mitochondria, and this is the cytochrome C. When Puma, Noxa, BH3 proteins bind with anti-apoptotic, they will sequester, and Bach's protein is free. Bach's protein come here and bind with Bach protein. They form channel, and the cytochrome C, which is in the intermembrane space, is going to the outer surface of the mitochondria. Now cytochrome C, apoptotic protease activation factor 1, APAP1, is added here. That means APAP1 factor is added with the cytochrome C. Now cytochrome C will be bind like that. And now come to the point second, the cytochrome C and the main APAP1, the receptor 1, APAP1, there is a CAR domain, Casper's recruitment domain, in short, CAR domain. Okay, this CAR domain, after binding the CAR domain, there will be the binding of procaspase 9, 7, 3, 3, 6 and 7. 7 cytochrome C with APOP1 with binding there together, and with then procaspase 9 will bind with them. Okay, so they form a complex structure with APOP1 cytochrome C and APOP1 7 APOP1 7 cytochrome C and 7 procaspase 9, and they break the procaspase into two types, a larger one and the smaller one, and they bind together become like this. That means procaspase break together to form caspase 9 actively. We come to the structure of the caspase. There is the color domain, caspase recruitment domain, where the caspase will be recruited. Pro domain. This is the actually large portion, large domain. This large domain, procaspase, and the small domain. When this will break, the activation of the caspase is done. Okay, and they may go to the apoptosis, and the cell become apoptosized. Come to the extrinsic pathway, that is which I will discuss about the. This is the extrinsic pathway. Is the receptor, first receptor, and when the first receptor, we'll discuss about the structure of the receptor first. There is the FAD domain, FADD, that means fast associated death domain. Okay, first receptor, first ligand is bind with first receptor fast. Then what will happen? Then this type of procaspase 8 or 10 active here. Recall that caspase 9, procaspase 9 is activated in intrinsic pathway. Now come to the extrinsic pathway. Here the procaspase 8 or 10 is active. Okay, so here the procaspase 1 they have a FAD domain and death effector domain. This is so when first ligand is bind to the first receptor, FAD domain, that means fast associated death domain, is added with FAD adapter protein, which have death effector domain. Death effector domain is bind with here, and death effector domain can recruit the procaspase 8 or 10, like that. Okay, and the procaspase is activated to the caspase 8 or 10, and they can help in apoptosis. The total complex is called DISC, means death inducing signal complex. The actively formed procaspase 8 or 10, they can cause the apoptosis, so it initiates the apoptosis process. So DISC is death inducing signal complex. Okay, in some insects, IAP, that means inhibitor of apoptotic protein, is present, which can stop apoptosis, and obviously there are some anti-IAP, that means inhibitors of apoptotic proteins are present, like RIP, HID, GRIL. In human, generally SMAC, Diablo, OMI, that's are present. All IAP, that means inhibitors of apoptotic proteins, have one or more bead, that means baculovirus repeat protein, baculovirus IAP repeat. Okay, so BH3 only protein are the largest subclass of BCL2 family proteins, because BCL2 protein generally, what happened there? We discussed about that beforely in short, that BCL2 protein, the BH3 domain, BH3 only protein, which bind with the anti-apoptotic protein and destroys them to free BAC and BACs, that means pro-apoptotic protein, which will be take part in the apoptosis. Okay, so this is the apoptosis. There are some points that many cells produce inhibitory proteins that act either extracellularly or intracellularly to restrain the intrinsic pathway. For example, some produce cell surface decoy receptors which have a signal binding domain but not a death domain, because they can bind a death signal but can't activate apoptosis. Okay, the decoys competitively inhibit the death receptors. Decoys competitively inhibit the death receptors. Cells can also produce the intracellular blocking proteins such as FLIP, which resembles an initiator procaspase but lacks the prokaryotic domain, which competes with procaspase 8 or 10 for binding sites in the disc death inducing domain, which I talked before in the extrinsic case. And they thereby inhibits the activation of these inhibitors, initiator procaspases. Such inhibitory mechanism help prevent the inappropriate activation of the extrinsic pathway of apoptosis. In some circumstances, death receptor activate other initiators, other intracellular signaling pathways that do not lead to apoptosis, that is TNF receptors. For example, they can also activate the NFKV pathway, which can promote cell survival and activate genes involved in the inflammatory responses, which response is the dominant depends on the type of cell and the other signal acting on it. Okay, so this is all about apoptosis, and generally one type of question you can get obviously in net, CSIR net, from here. Okay, so prepare it very carefully. Thank you very much. If you like my video, then please like, subscribe and share my video. Thank you.